This is our presentation for the midterm project review by Josh Narrow, Nick Forzano, and Sean Egan. And we did chapters 14 through 19. Mendel performed the pea experiment to study inheritance. He used peas because they have a lot of variety. Mendel also came up with the law of segregation. The law of segregation includes genes account for variation. Organism inherits two copies of a gene, one from each parent. One is dominant and one is recessive, and the alleles segregate during gamete formation and end up in different gametes. Monohybrids are heterozygous for one particular character, and dihybrids are heterozygous for two characters. A dominant allele is fully expressed in the phenotype of a heterozygote, and a recessive allele is not. Homozygous is two identical alleles. Heterozygous is two different alleles. Phenotype is an appearance. Genotype is the genetic makeup. Punnett squares are diagrams that predict possible allele compositions of offspring. Law of independent assortment is each pair of alleles segregates from other pairs of alleles during gamete formation. Complete dominance is when only one allele is shown. Incomplete dominance is when both alleles are shown with a mix. Codominance is when both alleles affect the phenotype in separate, distinguishable ways. Pedigrees are charts that show phenotypic traits collected from many individuals in a family over many generations. Fetal testing is when they test to see if the fetus has a certain genetic disorder. Thomas Hunt Morgan was the first to associate a specific gene with a specific chromosome. He found this by experimenting fruit flies. Chromosomes carry genetic material when they are found in the nucleus in the eukaryotic cell. In mammals, if an organism has two X chromosomes, then it's a female, and if it has an X and a Y chromosome, then it's a male. Both sex chromosomes segregate during meiosis. Individuals with an SRY gene develop testes. A gene located on either sex chromosome is called a sex-linked gene. It is far more likely that a male will get a sex-linked disorder than a female. Chromosomes can be damaged by physical and chemical factors. Non-disjunction is when the meiotic spindles cause problems with the daughter cells. This is an error during meiosis that can alter chromosome number in a cell. There are 4 ways to change the chromosomal structure: deletion, duplication, translocation, and non-disjunction. Several human disorders are caused by mutations to the mitochondrial DNA. Frederick Griffith found out a transforming factor can change phenotype. Chargaff is the one who put together adenine and thymine and cytosine and guanine. Hershey and Chase confirmed DNA is a genetic material. Watson and Crick, with the help of Rosalind Franklin, confirmed DNA has a double helix structure. Messelson and Stahl created a semi-conservative replication. DNA replication is when DNA creates a copy of itself. DNA replication uses many enzymes to replicate, as shown here. The central dogma is the flow of genetic info in a cell, DNA to RNA to proteins. DNA is like a ladder. The 4 bases form the rungs, and a hydrogen bond is between them, and the backbone is made of phosphates and sugars. The strands run from 5 to 3 prime. Complementary strands run in opposite directions. There are 4 bases: adenine, thymine, cytosine, and guanine. Purine is a double-ringed, and adenine and guanine are purine. Pyrimidine is single-ringed, and that is thymine and cytosine. Adenine goes with thymine, and cytosine goes with guanine. DNA replication has leading and lagging strands. Leading strand is synthesized continuously, and the lagging strand is Okazaki fragments joined together by DNA ligase. Each strand serves as a template for a new strand. Enzyme helicase unwinds double helix of DNA, which is stabilized by single-stranded binding proteins. DNA polymerase III synthesizes leading strand. It adds bases to the 3' end of the growing DNA strand. For bases to be added, it needs RNA primer, which is created from the enzyme primase. DNA polymerase I removes RNA primer and replaces it with DNA nucleotides. Telomeres are repeating non-coding sequences at the end of the chromosomes that serve as protection for the chromosome. Enzyme telomerase extends telomeres and can add DNA bases at the 5' base. DNA polymerase proofreads and corrects errors and removes abnormal bases. RNA has ribose sugar. It has uracil instead of thymine, and it is single-stranded.
Transcription makes mRNA from DNA, and RNA is built in a 5' to 3' direction. Transcription initiation. RNA polymerase binds to DNA at the promoter. It separates DNA strands to begin transcription. Elongation. The RNA chain grows in a 5' to 3' direction. Nucleotide bases pair with template strand. Non-coding strands of the DNA reform the double helix by pairing with the coding strand. Termination proceeds until RNA polymerase reaches a terminator site on the DNA. The RNA molecule is then released. The TATA box binding site is for RNA polymerase and transcription factors. In eukaryotic cells, there are introns and exons. Introns get spliced out by spliceosomes, which are made of SNRPs. A single base added or deleted causes a mutation. At the 5' end, a GTP cap is added, and a poly-A tail is added to protect the mRNA when it enters the cytoplasm. Translation initiation starts when a small ribosomal subunit attaches to the 5' end of the mRNA. Start codon, AUG, methionine. Elongation, ribosome moves along the mRNA attaching tRNA's anticodon with the mRNA codon. tRNA then attaches with the new amino acid at the A site, then adds the amino acid to the growing polypeptide at the P site. Before exiting at the E site, a new tRNA enters the A site and repeats the process. Termination occurs when ribosome encounters a stop codon, ribosome subunit detaches, and the polypeptide chain is released. A point mutation is a single base changed or substituted. This can cause a nonsense mutation, which is a change in the stop codon. A missense is a change in the amino acid. A silent mutation is no amino acid change, but is a redundancy in the code. Viruses are infectious particles consisting of a nucleic acid in a protein coat. Capsids are protein shells that enclose the viral genome. They are built of protein subunits called capsimerase. Viral genomes consist of double or single-stranded DNA or RNA. Some have membrane envelopes that help them to infect the host cells. Viruses are obligate intercellular parasites, meaning they can only reproduce in the host cell. Each virus has a host range, meaning there is a limited number of host cells it can infect. The lytic cycle results in death of the host cell. There are 5 steps: attachment, injection, hydrolyzation, assembly, and release. In the lysogenic cycle, the genome is replicated without destroying the host cell. Viral DNA is incorporated into the DNA of the host cell. Retroviruses are viruses that can transcribe DNA from an RNA template. They use reverse transcriptase.
And that's our slideshow. Sorry, Miss Oma, we can't read.